Okay, welcome to week 5 and 6 and 1 PS pain relief. You guys have just come back from your mid-sem break, I guess, so hopefully things have gone well, you've had time to relax, revise, all that good stuff. For today's lesson or session, we've got connective tissue, immunology, and neurophysiology we'll be covering. So just to start off with, I guess I just wanted to remind you guys, if you ever have questions, know that you can ask us. It'll just be, through Facebook, through our Google form, whatever. Just make sure you get in touch. And happy studying. So starting off, we've got connective tissue. We'll be covering, what is connective tissue. We'll be talking about its composition, classification, and then finishing off with a bit of talk about cartilage and bone. Just on this right-hand side here, we'll end up talking about areolar tissue, adipose tissue. You've got dense connective tissue, cartilage, bone, and blood. And this is just kind of the graphic representation of what histologically or under a microscope, what these tissues would look like. So what is connective tissue? Connective tissue supports, connects, and separates different types of tissues in the body. So you've got a couple of examples. We've got blood, which we can consider as a loose connective tissue. We've got bone, cartilage, adipose, a bunch of different types. You can see them on the right-hand side here, what they look like. And embryologically, connective tissue arises from ectoderm and mesoderm. They're composed of ECM, extracellular matrix, so extracellular, outside of the cell, it's a matrix, right? And that's composed of fibers and ground substance. First of all, ground substance is a liquidy fluid gel matrix, has nutrients and other things dissolved in it. And you've got those fibers. Two main fibers I want to talk about, one being collagen and the second one being elastin. Collagen you've probably heard of before. It's the main fiber and it's the most abundant in the body. It provides tensile strength, so it's like a structural protein, Then we've got elastin. So elastin is found as fibers or as a sheet, and like the name suggests, it's a type of fiber which provides elasticity. So just if we have a look at this photo on the bottom right-hand corner, you've got the collagen fibers and the elastic fibers, those dark purple elastic fibers. The other component in connective tissue is the cells. So just down this bottom here, I've got, sorry about that, I've just got examples of the different endings for cells. So in the same way how we learned that enzymes end in A's, right? Amylase, lipase, etc. We learned that the endings can often indicate what a cell does. So if a cell is a blast, it synthesizes and secretes. A site is a mature cell. Clast is the type of cell which breaks down other bits. So the kind of sentence you use to memorize this is blasts build, sites sit tight, and clasts clear. Got some examples on the left-hand side there with fibroblasts, which synthesize and secrete collagen. Really important cell. You've also got things like adipocytes, storing fat, osteocytes, which maintain bone, and chondrocytes, which are there to maintain cartilage. Alrighties. Next thing we've got is the classifications of different cells. So on this left-hand side, we've got a bunch of different things, all these images, but the really key thing I want you guys to focus on is the loose and dense, just on the right-hand side. So the classification of loose connective tissue is that it's flexible, there's a network of fibers with many open spaces. So we're talking about fat and areolar tissue. And you can see that just up top, top left-hand side, fibroblasts, ground substance, and protein fibers. You just whip out this pointer. This pointer here. Next thing we've got is that dense connective tissue. This one's got a couple types on it, but I kind of just classify it into irregular and regular. Dense connective tissue has a high fiber content, so a lot of collagen, a lot of elastin, and can even be hard if it's mineralized. So in this example, I'm talking about bone. Bone is dense connective tissue which has been mineralized. In contrast, you've also got, the dense irregular tissues. These are the kind of tissues which can resist tension in all directions. if you guys think back to when we did epithelium, we talked about transitional epithelium, which is found in the bladder, right? And it can stretch in all directions. Same thing with skin, same idea here. You've got the irregular arrangement of cells in all different planes. So example here is that dermis layer, so that deep layer in skin. You've also got the regular connective tissue. These are the ones which have fibers aligned linearly, so they're all parallel and resist force in one direction. So whether it's collagenous or elastic makes a small little difference in terms of elastic ones being able to, stretch and recoil like a large blood vessel, versus the collagenous ones, really common in tendons and ligaments, strong in one direction. Okay? If you want to have a look through the rest of this photo, you've got the reticular and adipose. Adipose is just that storage of fat, warmth, insulation. Reticular connective tissue is the kind of tissue you'd find in a lot of organs, so inner organs. Okay? Next thing, epithelia. Oh, sorry. Just a diagram pulling together epithelia and what we're learning today with the connective tissue. On the left-hand side, we see that simple squamous, simple cuboidal, simple columnar with some nice histology showing also next to like the organs that they can be found in. It's just a nice little example. On the right-hand side, we're seeing some of that new stuff from today. So that connective tissue in terms of, dense fibrous connective tissue in ligaments and tendons. See that linear direction. You see the collagen fibers, and obviously a couple of fibroblasts and things like that. You've also got areolar tissue, lots of spaces in between them. You've got elastic and collagen fibers. And then that adipose tissue at the bottom, storage, big vacuoles containing the fat droplets. Okay, next, cartilage. So cartilage has a specialized extracellular matrix, and it's made by the chondrocytes. What's also important to talk about and note for cartilage is that it has no nerve, lymph, or blood supply. So sometimes you'll hear that it's avascular, without vascular vessels. And that we'll talk about that just over the next slide, but that will impair or limit the ability of cartilage to repair itself, to regenerate. Three main types you've got to know about. You've got hyaline, elastic, and fibrocartilage. These are just some descriptions in terms of how you can see them histologically. You've just got them on the right-hand side as well. Hyaline cartilage is the one which reduces friction. So we're talking about surfaces which are moving, bones articulating at joints against other bones, right? Elastic cartilage is like the weird ones, like your ears. They can distort, they can move a little bit. They're a little bit more flexible, they're elastic. And last of all, that fibrous cartilage, there for that shock absorption and resists compression. So you can see it, for example, in the spine. Okay, perfect. Next, here's where we're going to talk about bones, the ability to heal, and a couple other things. So bone does have a really good neurovascular supply, which allows it to regenerate so well. If someone had arthritis, that doesn't really heal because cartilage can't really heal itself. Whereas if we're talking about, someone breaking a bone, they can heal, they can heal in like not a long time at all, couple of weeks even. That's really fast, and that's because of that ability to re-heal is because of that blood supply. What that blood supply does is it means that cells are able to migrate and repair. So you've got osteoblasts and osteocytes. What happens during the repair process is osteoprogenitor cells will differentiate into osteoblasts. Osteoblasts will secrete that calcium phosphate matrix, which is found in the bone, and they'll help that strengthen. And you'll get those osteocytes moving in, and they osteocytes are the mature cells. They're inactive osteoblasts trapped in osteons. So you'll find that in the roomy bones. Again, poor blood supply, cartilage doesn't regenerate well, so chondrocytes aren't actually able to migrate and replace damaged cartilage very well, and instead, cartilage is actually replaced often with scar tissue, so a different type of tissue. Okay, next bit, 3 segments of bones. I want to talk about the epiphysis, diaphysis, and metaphysis. So the physis, just right in between is the growth plate. Sometimes when kids fracture their growth plate, they'll have like stunted growth for a little while. It's not that bad, to be honest. But epiphysis is the ends of the bone. Metaphysis is where it's curving, so it's at that angle. And diaphysis is like the length of the bone. I'll also be talking about the next slide as well about the layers of bone. So we've got the periosteum, dense compact bone, endosteum, and spongy tissue, or sometimes called trabeculae. I've got a diagram over the next slide. We'll just go over and talk about some of these ideas again. So just to reiterate, on this left-hand side, we've got the diagram with the epiphysis, the end of the bone, metaphysis, the slanted part, and the diaphysis, the length of the long bone. You can also see, actually, there's the epiphyseal line right here, and that's often where bones would break because it's not as strong there. You can see the articular cartilage at the end, and it'll tell you, there's compact bone right here. Let's go through, look through this right-hand side now and talk about the layers of bone going in. So starting from the outside, you've got a periosteum. What that is a membrane, and it just surrounds the bone. It's collagenous, it has blood vessels, provides nutrients, and it has the osteoprogenitor cells, so the ones which are really helpful in bone repair. Then one more layer in, you've got that dense, compact bone. What we're finding here is sheets. It's organized in sheets. You can see those layers around like an onion, and it has calcified cylindrical osteons. You've got osteocytes trapped inside these osteons, and they're hard, they're strong. We go in one more layer, and we've got the endosteum, and you can see the neurovasculature going through here. Don't worry about that canal, but yeah, got the odd endosteum one layer in. Just like the periosteum, it's kind of a membrane, and this one lines the spongy bone. In the spongy bone, this is where you find that network of mineralized tissue. So you've got bone, you've got like the marrow and fat. It's often breaking and reforming a lot, and it's hematopoietic, which means it has a role in forming young blood cells. And just at the very bottom of this slide, we've also got a bit of bone histology. I just wanted to let you guys see and appreciate the difference. You've got osteocytes trapped in osteons, which clearly have layers to them. We've also got on the very left-hand side, just to appreciate the difference of how the bone marrow, hematopoietic bone marrow, will look so much different to the dense bone. And that's kind of the key thing for bones, and that wraps up connective tissue. So over the next slide, we'll have the start of immunology.
All right, so we'll move on to immunology now. So the first thing I want to talk about is the lymphatic system's role in immunity. So your lymph system absorbs and drains fluid between cells that has leaked out from the blood vessels, and lymph fluid is clear. So you have small capillary-like structures which lead into larger lymph vessels, and then finally they drain into lymph nodes. Keep in mind that this is separate from your vasculature, so it doesn't contain red blood cells. And lymph vessels also have valves to prevent backflow. Also, the heart doesn't pump the lymph system. It's your muscles and the thoracic movement that provides the pressure to pump lymph around the body. So some key things to note is that the net movement of fluid is from the tissues to the lymphatics and back to the blood. So the lymph vessels will eventually drain back into the vasculature. And then cells and material that are in the tissues will generally move along this pathway. So this feature of the lymphatic system allows our body to carry out immune responses because lymphatics carry immune or white blood cells, okay? So the reason why our body has can carry out immune function is because of the movement of fluid in the lymph vessels. So in part of your lymphatic system, you have your primary lymphoid organs, which is where immune cells are made, and that includes your bone marrow and thymus. And we'll talk a bit more about the bone marrow later. And then your secondary lymphoid organs are where adaptive immune cells, immune responses are generated. So this includes your spleen, peripheral lymph nodes, tonsils, etc. And you can see in this diagram here where the locations are. Okay, so your cells of the immune system they all originate in the bone marrow from hematopoietic stem cells, okay, so HSCs. So you can just study this diagram here. You've got your stem cells in bone marrow, and then they will either become myeloid cells or lymphoid cells. And so these are your group of myeloid cells, these are your group of lymphoid cells. Okay, so your innate immune system. So immunity can be described as having 3 different levels. First of all, you have your physical barriers, which block, disarm, or flush away pathogens that attempt to enter the body. So these include things like your tears, your eyelashes, all of which are designed to, first of all, keep pathogens outside of your body. If they do, however, get into your body somehow, then your innate immunity will deal with these pathogens. So your innate immunity is your rapid, non-specific killing of pathogens that do make it into the body. And then finally, we have the highest level of immunity and the strongest level of immunity, which is adaptive immunity. And this is your long-term, specific, specialized killing of scarier pathogens. So innate immunity, we'll talk about how it uses pathogen-associated molecular patterns, or PAMPs, to recognize pathogens. And specific cells are involved with the innate immune system, which include phagocytes, such as macrophages and neutrophils that engulf pathogens, natural killer cells, or NK cells, that kill damaged self cells, and eosinophil degranulation for large pathogens that cannot be engulfed. And then finally, we'll talk a bit about the complement or the soluble system that directly kills pathogens and recruits innate cells. Okay, so pathogen-associated molecular patterns, or PAMPs, these are unique structures that are inside or on the cell wall, cell membrane of the pathogens, and our innate immune cells can actually recognize these PAMPs. And the reason why PAMPs are a great method of recognizing pathogens is because PAMPs are usually important for the pathogen's survival. So the pathogen isn't going to just evolve one day to not have them anymore, because they need PAMPs to survive. Okay, so our innate immune cells can recognize them, and these PAMPs include flagellin, which is on bacteria, or the lipopolysaccharide layer, again on bacteria, or double-stranded RNA, which is viral. Okay, so our innate immune cells have specialized receptors called pattern recognition receptors that recognize PAMPs, so PRRs. And there are different groups of PRRs. For example, toll-like receptors. Okay, so there are 10 types of toll-like receptors in the body, which exist on the cell surface or intracellularly. Okay, so you can see here you've got TLR4, TLR5 on the cell surface, whereas TLR3 is intracellular. So all of these toll-like receptors, they have similar structure but different specificity. So for example, your TLR3s will bind to dsRNA and are intracellular. TLR4 will bind to lipopolysaccharide and are on the cell surface. Oh, by the way, dsRNA is just double-stranded RNA. And TLR5 will bind to flagellin, and again, they're on the cell surface. Now, talking about macrophages and phagocytosis, so PAMP recognition actually triggers phagocytosis. So macrophages and neutrophils are both cells of the innate immune system that specifically carry out phagocytosis. And this is just a mechanism by which immune cells engulf or ingest pathogens into specialized compartments, which is called a phagosome. So it's basically, they engulf it, so your immune cells will engulf it, and then it will join with a lysosome, which will create a phagolysosome. And then you'll have the release of mediators to break down pathogen molecules. So these can be enzymes, free radicals and oxygen radicals, or hydrogen peroxide, which will break down the ingested pathogen. Okay, so I'm going to explain this diagram. It looks very chaotic, but we'll take it in steps. So this is just explaining the migration of immune cells around the body. So how do different immune cells get around the body to initiate an immune response? Okay, so remember we're still talking about the innate immune system here. And this is the top part is your blood vessel, and the bottom part is your tissue. Okay, so first of all, let's say we have the initial exposure to the pathogen, and you have the recognition of the PAMPs on the pathogen by your pattern recognition receptors on macrophages in the tissue. Okay, so you can see here the macrophage is this green blob. It will read the PAMPs on the pathogen. Okay, it's in the tissue. And then 2, the macrophage will respond by releasing cytokines. Okay, so here the little orange things are your cytokines, and will also release chemokines, which are these kind of like yellow triangles. And these will bind to receptors on endothelial cells in the blood vessels. Okay, so inside the blood vessels, you have endothelial. You will have endothelial receptors, which will bind to the chemokine that is released. Okay. 3. You'll have increased expression of surface adhesion molecules on endothelial cells. Okay, so you'll have increased expression of things like selectins, integrin ligands, and proteoglycans, all on the surface of endothelial cells. 4, your circulating leukocytes or your white blood cells will express as well selectin ligands, which will bind to the selectins, integrins, which will bind to the integrin ligands, and chemokine receptors, which will bind to the proteoglycans. So you can see here, this is your leukocyte. You've got, you can see that it's expressing integrin and selectin ligands and chemokine receptors, okay? All right, step 5. So more adhesion molecules means that you'll have increased binding of leukocytes to the endothelium. Okay, so you can see that here. They've come down because they're expressing more receptors, and because endothelial cells are also expressing more surface receptors, there's going to be increased binding between the leukocytes to the endothelium. And then these leukocytes will begin to roll along the surface of the endothelial wall of the blood vessels. And they also bind to chemokines. Okay, so you can see binding of chemokines here. And yeah, it's just rolling along. So then 6, your integrin binding goes from low affinity to high affinity. So your leukocytes will adhere a lot more firmly. Okay, so you can see your integrin is like this aqua color. 7, you have a step called extravasation, which is when your leukocytes respond to the chemokines on the endothelial surface, and they will actually move through the gaps in the endothelium into the tissue, okay, so that they can get towards the site of injury or wherever the pathogen is, okay? So your leukocytes will follow the chemokine gradient towards the pathogen to assist in clearance. And that's it. Okay, so move on to talking about the other different cells of the innate immune system. So I mentioned before your natural killer cells or your NK cells. So some pathogens can actually hide from phagocytes inside our own cells, okay? So therefore, engulfing them is not really an option. So what your natural killer cells can do is that they can detect infected or stressed cells and kill them to stop the replication or spread of the pathogens. So the way they do this is because normal healthy cells will give inhibitory signals that inhibit the NK cell from destroying it. Okay, so you can see in this little image here, your normal cell, it releases a signal that communicates to the inhibitory receptor on the NK cell, and because of that, it will inhibit the NK cell from destroying the normal cell. Infected cells, however, they don't give off this inhibitory signal anymore. And because of that, the NK cell can proceed to destroy the infected cell. So your eosinophils and degranulation. So some pathogens are too big to phagocytose, okay? So they're too big for macrophages or neutrophils to engulf them. So your eosinophils are cells that have a lot of granules inside them that contain toxic agents, such as proteases. And these cells will release their toxic granules when in contact with a pathogen, hence damaging the cell wall of the pathogen, okay? So this is a histological image, and these bigger purple structures with like a deeper purple inside is your eosinophil, and those are the granules, dark purple. Okay, finally, we'll talk about the complement pathway. So soluble complement proteins are released by the liver and they remain in the bloodstream. They can also recognize pathogen surface proteins, and once they recognize them, they will proceed to coat the pathogen with more complement proteins, which ultimately makes the pathogen easier to phagocytose, which is called opsonization. They can enhance local inflammation, which will attract more immune cells to the site of infection. And they can create a membrane attack complex, which will puncture the cell membrane of the pathogen and eventually leads to cell lysis. So you have different groups of complement proteins, all right? So there are over 30 types of complement proteins. And different groups will lead to different complement activation pathways. So you either have either the classical pathway, the alternative pathway, or the lectin pathway. All you need to know is that all of these pathways lead to the same outcome, okay, which is the activation of complement protein C3 to form C3A and C3B. So here's just a diagram which shows it a bit easier. Okay, so remember all pathways, your classical pathway, alternative pathway, and lectin pathway, they will all lead to the activation of complement protein C3. Okay, so see how it's all leading to activating your complement C3? And then the activation of C3 will lead to the formation of C3A, which is involved in inflammation here. C3A inflammation. And C3b, which is involved in opsonization and phagocytosis. So here, that's what it says here. So see how it's broken down into C3b and C3a. And yep, your later steps will be creating the membrane attack complex, which will lead to lysis of the microbe. Okay, so the inflammasome. So inflammation can also be triggered by tissue damage, not just infections only. And your inflammasomes are structures that are present within myeloid immune cells only. So your macrophages, neutrophils, and eosinophils, and they're triggered by DAMPs. So DAMPs is similar to PAMPs, but instead of being pattern associated, it's damage associated. Now, when the inflammasome is triggered, it promotes inflammation by releasing cytokine interleukin-1. So all you need to see here is that, so PAMP-driven inflammation, you'll have the involvement of a pathogenic bacteria, which will which will be recognized and then lead to inflammation to try and get rid of it. Or it can also be DAMP-driven, which is associated with tissue damage. Okay, and that's it.
Hello, Jaffies. Today I'm going to be talking to you guys about adaptive immunity. We're going to be talking a bit about what adaptive immunity is compared to innate immunity, and essentially what the crux of that is that adaptive immunity responds to specific pathogens, as in your body targets a specific pathogen that has already infected you. As a result, we'll see that adaptive immunity takes some time to develop, but it also gives you long-term protection. There's two types. So we have a humoral immune response and a cell-mediated immune response. And there's also three sort of important cells, including two very special lymphocytes, which we'll discuss in detail. So adaptive immunity can be given to us in two possible ways. The first is via vaccination, which gives us artificial active immunity. And the second is by actually getting the disease. And we call that natural active immunity. And we'll talk exactly about how this works shortly. So basically what's happening in adaptive immunity and in an adaptive immune response is that first we get a pathogen inside us, and our phagocytes, which you've learnt about, specifically our antigen presenting cells, cells that present antigens to other cells on their surface, those guys will eat the pathogen. They'll phagocytose it. They digest it inside themselves, and then they take those antigens, which are parts of the pathogen, and they display them on their outer surface. Then after that, your phagocytes, which have those antigens now on their surface, show them to adaptive immune cells, those T and B cells we were talking about, and they have to show them to a specific T cell or a specific B cell, which targets specifically that antigen. So each individual T cell or B cell targets a specific antigen, and that is predetermined at the birth of that T cell or B cell. It will only ever target that antigen. And once that antigen presenting cell has displayed that antigen to the specific B cell or T cell, that adaptive immune cell becomes activated, starts dividing, and also will do other special roles, which include releasing cytokines and producing antibodies. We can kind of see a short summarized diagram here. I'd like to point out a couple of things. We've got cytotoxic T cells here. They're talking about cytotoxic T cells killing germs. Now, they actually kill infected host cells, not germs. So we'll talk about how that works as well. And also, we can see actually, that was all I had to say about that one. Cytotoxic T cells kill our own cells. All right, let's have a look at our adaptive immune cells. We've got three main types to fill out the roles of this story. We've got our antigen-presenting cells. Those are the phagocytes I was telling you about. There's a few main types. The first two are the most important, dendritic cells and macrophages. But it's worth noting that B cells may also be professional antigen-presenting cells. So these guys have a few key features. One of those is a class 2 MHC marker, and we'll learn about what all that means. Their job is to eat pathogens and display bits of them on the surface. Then we've got our T cells, two main types, helper cells and cytotoxic cells. Helper cells, their main job is to release cytokines to activate the rest of the immune system, but they also have a role in delivering antigen parts to, say, our B cells for further activation. Then we've got our cytotoxic T cells, and as I said before, they're cells that actually kill our host cells when they've been infected, so that the pathogen no longer has a vehicle to infect the rest of your cells. Now, when we get infected, our body also forms a whole bunch of memory T cells and memory B cells. These guys are designed to stay in your system for a long, long time after infection, so that they're just like sentinels that wait for reinfection, and then they divide like crazy and launch a much bigger and much more rapid immune response. And hopefully, if you have lots of memory cells, that means that you actually don't get sick the next time you get the pathogen, and it gets eliminated by the immune system before you get sick. Finally, we have our B cells, two types, plasma B cells and memory B cells. Already spoken about memory B cells. Plasma B cells produce a whole bunch of antibodies, and you can actually see some of those antibodies in this little drawing here. So let's talk a little bit more about what those antibodies kind of do. So we've got a picture of an antibody here. This is sort of the basic shape they use to describe antibodies. We can see they've got a constant region and a variable region, and two antigen binding sites. Those are the most important things about antibodies, is that they've got sites that bind to antigens. And this is why they are so important to immunity. What they do is the B cell that is specific to the antigen that has been presented to it produces only one type of antibody that is designed to bind to that antigen. And that can do a whole bunch of important things. It can opsonize the antigen, which makes it easier for our phagocytes to eat them. It can neutralize them. So if that antigen is a crucial molecule for the infectivity of the pathogen, and then an antibody binds to it, it can stop that protein from doing its function and therefore stop disease. We have agglutination that clumps pathogens together so that they can be destroyed en masse by our phagocytes. And finally, they have a role in complement system. I believe Christine has already spoken to you a bit about complement. We'll talk about the specific pathway involved with antibodies. Let's talk about B cells. So these guys are the ones mainly responsible for producing antibodies. And you can see they do this by a few key receptors. So first, we have our B cell receptors. You can see from this diagram, they're shaped exactly like antibodies. They are essentially surface antibodies. And like all antibodies and B cells, they are specific to one type of antigen. When that antigen binds to them, the activation of the B cell begins. However, there's two steps to the activation of a B cell. The first is antigen activation, and the second is activation by cytokines. And these cytokines usually come from a T helper cell, or also known as a CD4 plus cell. Now, once they do get activated, they start to divide like crazy. They divide to form plasma cells and memory B cells, and we call this process clonal expansion. That's because the cells that are produced are clones. They're exactly identical to the original B cell, and the antibodies that they produce are also the same antibodies. So as a result, you get a whole bunch of cells and a whole bunch of antibodies that are just designed to target one antigen. Now, the plasma cells don't live for very long. They just pump out antibodies at a rate of about 2,000 antibodies a second, so a lot, and they do that until they die. And their activity gets enhanced as they go by more and more cytokines being released from our T cells. And our memory cells, as I said before, they stay in the system. Now remember, those memory cells will still only be specific to the same pathogen that they stopped last time. So that's how immunity works. If you get chickenpox as a really young child, you'll probably never get chickenpox again. Although, as you will learn in probably semester 2, you'll learn that you can also get shingles later in life from the same virus via different pathways. Here we've got clonal expansion. So we can see that our cytokines are activating this B cell, and it differentiates to form more B cells, which then differentiate to form more B cells. So we get loads of plasma cells, which pretty much die. They're circled in red once they've served their purpose. But our memory B cells stay in the plasma for a long, long time. All right, now let's talk about that complement system I was talking to you about and how they interact with antibodies. Basically, what happens is you have this complement protein called C1, which binds to antibodies once they've bound to an antigen. And that starts to activate a part of the complement pathway, which produces this little circular pore thing down here. It's called the membrane attack complex. And that membrane attack complex is designed to attack the membranes of cellular pathogens. And then what happens is that water and solutes and all these kinds of things are free to move in and out of the cell membrane. And if there's enough of these pores that form on that pathogenic cell membrane, that pathogen will hopefully undergo lysis and thereby neutralize that threat. So it's worth noting that the complement system is primarily a feature of the innate immune system, but in this instance, it can also be a part of the adaptive immune response, or at least it's related to it. All right. Let's talk about the role of helper T cells. As I said, these guys, their main job is to produce a whole bunch of cytokines to hype up all other cells. So they can hype up each other with cytokines. They can hype up B cells, macrophages. They can also differentiate to form killer T cells or cytotoxic T cells. And their cytokines are crucial in regulating the responses of all of these cells. So how do they do it? Well, they have a couple of important receptors. The first is this CD4 receptor. The first is a T cell receptor. So that T cell receptor binds to the class 2 MHC of an APC, of an antigen presenting cell. We'll talk about what class 2 MHC is momentarily. But the point is they bind. The co-receptor to that is CD4, which is why these cells are often called CD4 plus cells. And once they do that, that activates these T helper cells. And the reason it activates them is because the MHC2, you can actually see it on this diagram, if it's to activate the T cell receptor, has to have a little antigen bound to it. So the antigen presenting cell has eaten a pathogen, it's displayed its antigen on the surface via its MHC2 marker, and then that MHC2 marker with antigen has bound to the T cell receptor with the CD4 receptor as a co-receptor. That antigen activates the T helper cell, causes it to divide into cytotoxic T cells and more T helper cells, and then release cytokines to activate all your other immune cells. Now let's talk about CD8 cells. Now, we said that CD4 cells are called that because they have CD4 as a co-receptor with other cells' MHC, specifically MHC2. CD8 cells are called so because they have a CD8 co-receptor which interacts with other cells' MHC class 1. So note that CD4 interacts with MHC class 2, and CD8 interacts with MHC class 1. We'll learn a bit more about that in a moment. So, infected cells are the main target for cytotoxic T cells. They come along once they've been activated, of course, by cytokines and by T helper cells. They bind to a infected cell's MHC class 1 using their T cell receptor and their CD8. And if they detect the antigen to which they are specific, they start releasing a whole bunch of cytotoxic chemicals designed to kill that infected cell. These include perforin and granzymes, and they're designed to trigger apoptosis. You can see a little diagram here. Now, they don't just kill infected cells, they can also kill cells that have, for instance, signs of cancer if their antigens are specific to those. Again, if the cancer antigens activate the cytotoxic T cell. And they can also do this to bacterially infected cells. Now let's talk about MHC. MHC stands for Major Histocompatibility Complex. Big word. All it means essentially is a marker displayed on most of the cells in your body to show your immune cells whether or not they're infected or whether or not an infection is present. So MHC1 is found on pretty much all cells that are nucleated. It's also found on platelets which don't have a nucleus, but for example, it's not found on red blood cells or erythrocytes. Its job is to present things to cytotoxic T cells, antigens to the cytotoxic T cells. Remember that CD8 is the co-receptor for this interaction. And once it does that, of course, if the antigen is detected to be foreign, then the CD8 cell will cause the host cell to undergo apoptosis. But if there is no antigen, or the antigen is a self-antigen, the CD8 cell will leave it alone. MHC class 1 is also checked out by these innate cells called natural killer cells, which detect abnormalities in MHC1, and they also have some other functions. MHC2 is only expressed on antigen-presenting cells, and only professional antigen-presenting cells. So you'll find MHC2 on dendritic cells, macrophages, and B cells. So the job of MHC2 is to present antigens to T cells and B cells. And, pardon me, the result of that is, of course, activation of your special lymphocytes and of the adaptive immune response. And remember that MHC class 2 interacts with the CD4 co-receptor. Now, we've got two types of immune response. We've basically already learned about them. It's just about understanding the difference. So the cell-mediated response refers to non-antibody responses. So we're talking about cytotoxic T cells. We're talking about T helper cells and their roles in killing infected cells and in hyping up other cells with cytokines. The humoral response is so named because it mainly occurs in the bloodstream. Your antibodies are released in your blood and in your humors. And as a result, they can circulate throughout the body. So the humoral response refers to antibody-related immune response. So in essence, what happens on the whole, in a little bit more detail than the first slide, is we get exposed to a pathogen, which gets phagocytosed by our APCs, and then they display parts of that pathogen on their surface. Those parts are called antigens or epitopes. Then your antigen-presenting cell meets a T cell. This T cell, if it's specific to the antigen, will differentiate into helper and cytotoxic T cells as well as memory cells. Now then your B cell will also detect the antigen and its activity will be initiated, but remember there's two steps to this. So our helper T cells come along and release a whole bunch of cytokines to make our B cells start to go bananas. So our cytotoxic T cells, so then our B cells differentiate rapidly. They form plasma cells and memory cells, and the plasma cells start pumping out massive amounts of antibodies specific to the detected antigen. Then our cytotoxic T cells, which have also been activated by cytokines, will go around and look for MHC1 markers with foreign antigens. And if they find those foreign antigens on MHC1, then they kill the infected cells. Then our phagocytes go around, and they're basically cleaning up the mess that the plasma cells and cytotoxic T cells create. So dead cell parts, pathogens, antibodies, all these kind of things get eaten up and digested. And this response, this whole cycle, continues until the pathogen is eliminated, or else. Basically, if this doesn't work, then you're not going to be very well at all. And then, of course, the final step is that our memory T and B cells stay in our body for a long, long time. They stay in circulation, ready to strike again in case of reinfection. But please note that steps 3 to 9 are basically all happening at the same time. It's not necessarily as neatly chronological as we've said here. That's all from me. There's a small note on microscopy here, which I won't go through. Please note that it's low yield. Hope you guys enjoyed.
I'm going to talk about neurophysiology. I'll be covering the first two topics while Mayan covers the last two. So I'm going to start off first with the resting membrane potential. So under resting conditions, the potential difference across cell membrane is negative 70 millivolts. So that's the inside relative to the outside. We set the outside at 0, and the inside is negative 70 compared to the outside. And this is generated due to differences in the concentration of ions, and these ions are charged. And this arises from 3 factors. So first, that the ions want to diffuse down the concentration gradient through the membrane, and they do so through channels. The second is the selective permeability of the membrane, and that depends on the number of channels or the number of channels that are open or closed. And lastly, is electrical attraction of cations and anions. So first we're going to look at the movement of potassium and sodium because they're the most important. So they move passively and actively, mostly passively. And like the passive part is more important than the active part, I guess you could say that. So for potassium, if you look at this table here, you'll see that potassium is more concentrated on the inside of the cell, so therefore through these leak channels or leaky channels, they'll flow out of the cell. And as for sodium, it's more concentrated on the outside, so it will try and flow down the concentration gradient and flow inside. And potassium has a greater influence on resting membrane potential than sodium, and this is because the plasma membrane is much more permeable to them than sodium, and it's more permeable. When we say more permeable, it means that the plasma membrane has a lot more potassium leak channels than sodium ones. And as for active, this is done through the sodium-potassium pump, also known as the 3 sodium 2 potassium ATPase. And they use ATP to actively pump 3 Na out and 2 K in. So this is in the opposite direction as the leak channels, so because it goes against the concentration gradient. And this is to make sure that the sodium concentration levels remain high on the outside and the potassium ion channels remain, the potassium ions concentration remain high on the inside. Yeah, so this counterbalances their passive leakage through the leak channels. So yeah, that's a pump. And now we're gonna move on to the negative particles. So that's chloride and A-. A- are negatively charged intracellular proteins. So chloride here is a principal ECF anion. Most cells are highly permeable to it, thus there's no active mechanism to transport. It can just diffuse across. So yeah, it passively distributes itself, and it's associated with sodium, obviously, because it's negatively charged and sodium is positively charged. And you'll see that the equilibrium potential of Cl is negative 17 millivolts. So it's already the resting membrane potential, but it doesn't really have an influence on the resting membrane potential at all. The resting membrane potential is mostly dependent on potassium and sodium concentrations. So rather, the resting membrane potential kind of influences where it goes in or out. Yeah. And so the negatively charged intracellular proteins, they're really big. So they're not permeable to the cell membrane, so all they can do is bounce against it and come back in. So it just doesn't go outside. Yeah, and they contribute to the net negative charge in the ICF. So Michael Leung mentioned some equations to calculate equilibrium potential. This is really not important at all. You'll note that I didn't even include the equations in the slide at all because it really doesn't matter. You just kind of have to know what they measure. So in an equilibrium potential, there's no net movement of ions across the membrane at all, but that doesn't mean there's no movement at all. There's just no net movement. And the first is I don't even know how to pronounce that, Nernst equation. So all you have to know is that it calculates the equilibrium potential for just one ion at a time, while the other one, the GHK equation, can calculate multiple ions at once. So if we look at just potassium, if we calculate the equilibrium potential for just potassium, it's negative 90. while for sodium it's positive 60. Yeah, and this is not that important, but I'm just going to go over it. So potassium concentrations are higher on the inside, so therefore it diffuses down its concentration gradient out. This makes the outside more positive, as now there's more positive potassium ions on the outside. But however, the negatively charged proteins on the inside can't diffuse out, so its negative charge draws the potassium ions back in, and this is known as an electrical gradient. And so these two gradients, the concentration gradient and the electrical gradient, when they balance out, what they give for the equilibrium potential of potassium is just negative 90. As for sodium concentration is higher on the outside, so it diffuses in down the concentration gradient, and then the ICF becomes more positive. But because the ECF has negative chlorine, and it has also lost some sodium, the sodium will diffuse out. That's the electrical gradient, and the two gradients balance at positive 60 millivolts. Yeah, right. So as we see with the other equation, that in reality, the membrane potential is negative 69 millivolts. And this is because potassium, like we said before, there are way more channels for potassium in the cell membrane, so it exerts a dominant effect. So we'll see that our real, true membrane equilibrium potential is way closer to negative 90 than positive 60. It's negative 69. So yeah, it's because potassium has more effect. And this, remember how the resting membrane potential is actually negative 70, and that's because the last negative 1 millivolt is contributed by the sodium-potassium pumps. Yeah. And now we're going to move on to the channels. So how a change in membrane potential is caused is that first there's a triggering event, and this changes the membrane permeability. So like I said before, that depends on all the channels, and if there's a change in the channels, of course there's a change in the ion movement across the membrane, and therefore that's how the membrane potential is changed. And so these water-soluble ions pass through the lipid bilayer only through these channels, and these channels are very specific to the type of ion that it allows to go through. So first is a leak channel, so there are potassium and sodium ones, and like I was talking about earlier, it's what's responsible for the resting membrane potential. So the thing about them is that there's nothing that stimulates them to open or close. They kind of just have an intrinsic rate of switching between open and closed states. And so yeah, like I said, it regulates the resting membrane potential and also contributes to transmembrane voltage of the excitomembrane. This is just a fancy way of saying that it can be like excited, I guess. And yeah, so there are 20% more potassium ones than sodium ones. We've been over this many times. So yeah, these are some photos of it here. So just randomly open, and the ions will flow down their concentration gradient. And now we're going to look at gated ones. So there are ones, well, where their opening or closing are controlled, and there are 3 different types. So the first kind is voltage gated. So this means that they open when the transmembrane voltage changes. So the charge of the inside relative to the outside, or vice versa, changes. And so so it's done because the amino acid in the protein is sensitive to charge. And they participate in the generation and conduction of action potentials. And you'll see that they have this like little plug that plugs it when it closes or opens. Yeah. And the next is mechanically gated ones. So a mechanical change can be something like changes in pressure, touch, or temperature in the surrounding tissue. And so I guess an example of this that you'll come across later is like mechanoreceptors. Like when you touch something, there's like a change in the tissue around it. So these gates open. And lastly is a ligand, or chemically gated ones, also known as ionotropic receptors. So a chemical messenger or ligand would be something like a neurotransmitter, a hormone, or certain ions, and they'll just bind to the outside and trigger it to open or close. Okay, and now we're going to look at electrical signals. So neurocommunication can be done electrically or chemically. And excitable tissues, there are 2 kinds in the body, and one is obviously neurons, so they receive, process, initiate, and transmit messages, and the other type is just muscle cells, and when they're excited, they contract. And there are 2 types of electrical signals. They're graded potentials and their action potentials. So graded potentials are short distance signals. They're local, small changes in the membrane potential. They're very short distance, only a micrometer to millimeter, and they spread bidirectionally. They spread in both directions, yeah. And they are produced by a very specific type of triggering event, and they can, but not always, initiate an action potential. And an action potential is long-distance signals, so they're brief, rapid, large changes in the membrane potential, so it could happen really quickly, 1 to 4 milliseconds, and the changes in the charge can be very big, up to 100 millivolts, and they're long distance. So, while it's micrometer or whatever, for a graded potential, these can go up to 1 meter. And a buzzword is kind of that they're all or nothing, so they either depolarize or they don't, and they propagate in only one direction instead of bidirectionally. And they start with a small portion of the membrane, but it's propagated throughout the whole neuron. So yeah. Oops. Okay, so we're gonna look at a graded potential first. So yeah, like I said, there are local changes in the membrane potential. The magnitude can range, and their uses is in things such as a postsynaptic potential, receptor potential, amplite potential, pacemaker potential, and slow wave potentials. This is not that important, I guess. So something important to note is that the stronger and longer a triggering event, the larger and longer the graded potential is, and it's because if they're stronger or longer, that there are more gated channels that open, and they're usually caused by a very specific triggering event that causes gated ion channels to open in a specialized region of the excitable cell membrane, so in just this particular region of a membrane. And sodium moves in, sodium channels open, and sodium moves in, and depolarization is confined to this small specialized region. And they're also spread by passive current flow. So the remainder of the membrane, besides the active areas, the resting membrane potential, and currents passively flow in both directions, in and out between the active and adjacent resting regions, so like on both sides of the membrane. And they die over a short distance, so they have a decremental spread. So the ions just leak through the open channel. So if you look here, this triggering event only triggers this like specialized portion of it, and sodium channels open and sodium ions flow in. So for a brief period of time, the inside becomes a bit more positive instead of more negative. Yeah, and this just slowly, it goes both ways, and it just slowly dies out, just slowly tapers out. Yeah. So now we're going to move on to an action potential. Action potential is a brief, rapid, large changes in the membrane potential that causes the potential to reverse the inside relative to the outside transiently, and they're non-decremental. Like I said, they're all or nothing. So once it happens down the whole neuron, and it doesn't die out. So, like I've already said like a billion times now, resting membrane potential, the cell membrane is 25 to 30 times more permeable to potassium than sodium. But during an action potential, this changes, and so the ions will flow down their concentration gradients. And an action potential can be initiated by a graded potential of sufficient magnitude. Yeah, we'll look at this more on the next slide. So first we're going to look at the channels that are important for an active potential to happen. And they're both voltage gated. So they're voltage gated sodium channels and voltage gated potassium channels. So the important thing to note is that for sodium voltage gated channels, they don't just have a closed and open state, but they also have an inactivated state. So normally they're closed at resting membrane potential, and when the cell depolarizes, they can go into an open state. And when they open, obviously sodium flows down their concentration gradient, and because they're more concentrated on the outside, they flow in. But they also have this inactivated period. So first they open, and then they become inactivated. So not only are they closed, but they can't even be open again, even if they're stimulated again, because they have this inactivation gate plug that like plugs the hole, like the opening of the channel. Yeah, and we'll see later that this is essential for something called the refractory period. And they also open and close very rapidly. As for potassium voltage gated channel, it's also known as a potassium delayed rectifier, or like KDR. And I guess it's quite important to remember this name because this is important to remember that they open like after a short delay from depolarization. So it's important to note that they have a delay, and they only have an open and closed state. So they're closed at rest, and they're when they open after depolarization, potassium ion flows out of the cell, because remember they're more concentrated on the inside. Yeah, so they open and close slowly. Potential propagation. So we're just going to go over the whole action potential and how it works. So first, of course, at resting membrane potential is -70, right? So all the voltage-gated channels are closed and only the leaky channels are open. Yeah. And then there's a depolarization. And so first there's the stimulus or triggering event that causes the local membrane potential to increase, like this. So when this happens, this causes some sodium channels to open. And when some sodium channels open, some sodium flows into the cell to further depolarize the membrane. And by doing so, they activate even more sodium channels. And eventually it reaches something known as a threshold potential. So it's quite important to know that this is around -50 to -55 millivolts. And once it reaches that, there's an explosive depolarization that happens, and the membrane potential goes up to +30. It's not negative, it's going to be positive. Sorry. And so, yeah. So there's a depolarization like that. And the reason that the potassium, not potassium, the reason that the potential doesn't get any more positive than this is because all these sodium channels then enter in an activated state. So they become closed. So basically there can't be any more flowing in, so it doesn't continue to get any more positive. Yeah, and it's important to note that during this time, the voltage-gated potassium channels are also opening, but slowly. And next, so there's the propagation. So propagation only travels in one direction from its initial site. So that's the axon hillock. That's right here. It only propagates one way down the axon. And this is due to something known as a refractory period. And during a refractory period, a new action potential can't be initiated. And we're going to be looking at this on the next slide. And next, there's repolarization. So at the point of depolarization, the membrane goes, the intracellular fluid goes all the way from -70 to +30 millivolts, right? But it will have to slowly return back to -70. So remember how while the voltage-gated sodium channels are open, the potassium channels are also opening, but they open more slowly. So after the sodium channels are already opening and closing, this is when the potassium flows out of the cell, because these channels are now open, and they carry the positive charge back outside. So first, when the sodium flows in, it brings positive charge inside, but then the potassium one's open, and the potassium brings a positive charge back outside. Yeah, and so this slowly restores the negative resting membrane potential. And normally there's a hyperpolarization period. So the sodium channels are reset, but the sodium channels are still closed, but the potassium channels still remain open because they're still closed, and it just allows like continued potassium ions to flow out to make it like make the inside even more negative than before. And once all these channels close, eventually the membrane potential of -70 will eventually happen again, and this is done by the actions of the sodium-potassium pump. So if we go back here, so remember when we said, it depolarizes a bit, reaches a threshold, then completely depolarizes, and so when it depolarizes, it's when the sodium channels open and sodium ions flow in. And then the potassium channels open a little later, and when they open, this repolarization, because the potassium ions are flowing out and bring the positive charge back outside. So it goes down, 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 but it continues to go down because the channels are not only slow to open, but also slow to close. Yeah, so it reaches here, and then the restoration back to the resting membrane potential is done by the active pump. Okay, so lastly is the refractory period that I mentioned before. So the refractory period is when, there's two different kinds. There's the absolute one and there's the relative one. So the absolute one means that there is no way that the neuron can depolarize again. And this is because all the sodium channels are currently in their inactive state. So there's no way they can be open no matter how you stimulate it. So yeah, and they can't be stimulated until the membrane potential returns to resting membrane potential again. Yeah, so you'll see this is the resting membrane potential, and until the cell membrane returns here, we're in the absolute refractory period. And the relative refractory period means that the sodium channels are no longer in an activated state, they're in a closed state, so technically they could be activated. But the threshold is originally here, but it's actually now higher, so it would take more, like the trigger event has to be greater for the membrane to depolarize. It just basically means that the threshold is higher, so now it's harder to depolarize again. And this is just because the potassium channels are closing, so the entry can still cause it, but yeah, it's phased. And this is because action potentials are actually generated by the movement of a very, very small fraction of total ions. So in reality, the overall concentration gradient of potassium and sodium are actually still maintained during action potential. So, yep, and that's it. We're going to move on to Mayan.
Okay, so now let's move on to the next topic in our series on neurophysiology. So the spinal cord, when this was taught to us, I kind of found that personally it wasn't super well organized, and so I'll attempt to make it a bit clearer for you guys. So how are we going to organize this? First, afferent/efferent neurons. These speak to the directionality of neurons, and they don't really say exactly what kind of path it follows, only that afferent means going towards and efferent means going away from. So if we have our arrows right here, you see one going towards and one going away. So synapses will join these neurons into ascending and descending tracks. So we have here again our ascending and descending tracks. And then we have monosynaptic and polysynaptic reflexes. So this happens if you want your reaction to be a bit faster than having to go from your sensory receptor to your spinal cord, to your brain, to your spinal cord, and then back out to your muscle. So a reflex arc would go simply just like that, there. So it's just very quick and takes place without involving the brain. And then last but not least, your physiological fight or flight or rest and digest responses. These are involuntary, and these come from your brain, and there we have it right there. That's a physiological response. So this is the concept diagram of what I'll be talking to you guys about. So once again, afferent/efferent neurons only speak to the directionality of the neuron. They don't say, well, they do. They don't say what specific type of stimulus they convey. Only that afferent neurons convey sensory information, and efferent neurons convey motor information. That's all we know, and that's all you guys need to know. So synapses are basically the junction between two or more communicating neurons. So the types of synapses that there are electrical synapses, which are just kind of tunnels that are between two nerve cells that are in contact, and these allow ions to pass directly from cell to cell. So as you can imagine, this is bidirectional, and it is also found in the brain, so it is very, very quick. All of this is very quick. Now, chemical synapses are slightly different in that they have a synaptic cleft, and the presynaptic neuron, which is the neuron that goes kind of first in the sequence, and then the postsynaptic neuron, which goes later in the sequence, they are not in direct contact. So they are separated by a synaptic cleft. So these neurotransmitters, they are going to be doing the work of transmitting the nerve impulse from the presynaptic neuron to the postsynaptic neuron. And this is how it happens. So just reading through the process very quickly. Once the action potential reaches the axon terminal of the presynaptic neuron, voltage-gated Ca2+ channels open. Oh, that's a bit of a grammatical error. Sorry about that. And then the influx of calcium ions caused by the opening of the voltage-gated Ca2+ channels, this will trigger the exocytosis of neurotransmitters. And then once these neurotransmitters are released, they will undergo exocytosis and diffuse across the synaptic cleft, binding to postsynaptic neurotransmitter receptors. And then the binding of these neurotransmitter receptors of the neurotransmitters to the neurotransmitter receptors will trigger the opening of voltage-gated Na+ channels, right? And as you guys know, an action potential begins when Na+ ions flow into the nerve cell and continue to depolarize the nerve cell all the way down. And so that's how you get the continuation of the action potential through the postsynaptic cell. But then how do you get, so what happens to the neurotransmitters that are left behind? Well, they're then degraded or taken up by presynaptic neurons and then recycled. So here, as you can see, this is just another helpful diagram for you guys, just in case you want a visual representation of synapses. And so next up, all right, I'll just move my frame up there. Okay, so spinal tracts. Now, what happens when your neurons join up to become pathways from either your sensory receptors or your muscles to your brain? Well, these become spinal tracts. So there are two types of tracts. There are ascending pathways and there are descending pathways. So the ascending pathways include the dorsal column medial lemniscus. I'm going to be calling that the DCML just because it's less of a mouthful. And this goes through the dorsal white column, which, as you can see right here, it's located on the dorsum of the spinal cord. So dorsum, remember that means posterior. That means on the back of it. And so it's located on the dorsum, and it's also located in the white matter. As you can see, this is an error right here. It should be white. It should be just the spinal cord. I'm going to fix that in the final edit of this PowerPoint slide. So they run through the white matter and they're located on the dorsum. That's why they're called the dorsal white column. Okay, and then the spinothalamic tract goes through Lyssaus fasciculus, and then eventually goes on to travel through the ventral and lateral spinothalamic columns. So Lyssaus fasciculus is right here. As you can see, it's kind of on this protuberance of the gray matter, and it will eventually go out to either this or this. All right, and then the descending pathways. This basically just includes the corticospinal tract, which goes from your brain through the lateral corticospinal tract and the anterior corticospinal tract. So as you remember from the previous, from the DCML, if this is the dorsum of the spinal cord, then this is the ventral aspect of the spinal cord. And if dorsal means posterior, it means the back, then ventral means anterior, it means the front, okay? And then here we have the lateral corticospinal tract. Lateral means on the side, and it's opposed to medial, which is in the middle. I'm sure you guys have probably heard this somewhere, but if you haven't, then now you know. And next up, if my computer would just work. Okay, yeah. So each tract has a specific function. So the ascending pathways, including the DCML and the spinothalamic tract, these carry sensory information. So the DCML takes care of fine touch, pressure, vibration, and proprioception. So all of these are kind of delicate. They're the fine kind of nuances of sensation. So that's why you remember it as the DCML is delicate. So D for DCML, D for delicate. And the spinothalamic tract takes care of crude touch, pain, and temperature, which, as we all know, are the spicier aspects of sensation. So the spinothalamic tract is spicy, and that's how you remember that bit. Now, the corticospinal tract, all you guys need to know is that it takes care of movements of the limb and trunk. So the corticospinal tract helps you crank it. I just made myself sound like 30 years old. Okay, okay, let's start with the DCML. So the DCML runs from your sensory receptors into your spinal cord, where it involves the dorsal column, which consists of the gracile and cuneate fasciculi. Oops, sorry. So what is a fasciculus? Okay, it's just a bundle of nerves. So gracile means long and slender. Cuneate means wedge-shaped. You can see the reasoning for that later on in this, if you just read through this yourself. But basically, this is the gracile fasciculus. The cuneate fasciculus. It carries nerves that run from your upper body and your trunk, and it's kind of wedge-shaped, and the gracile fasciculus carries information from your lower limbs, and it's a little bit more long and kind of slender looking. It goes up to so this first neuron goes through that to the medulla oblongata, which contains the medial lemniscus. So now you can kind of see the terminology beginning to take shape. So we have dorsal column, which is where these neurons run in your spinal cord, and we have the medial lemniscus, which is where the neurons run in your medulla. So this is part of your brainstem. So then we have the pons. Midbrain, thalamus, and postcentral gyrus. And your second neuron would have synapse in the medulla. It would go through all of this, and then it would synapse inside the thalamus, where your third neuron then takes over. It goes from the thalamus into the postcentral gyrus. So that's the overview of everything. And now let's start on a bit more detail. So basically, your first neuron goes from having delicate touch. So let's say with a feather on your hands and feet, your upper and lower limbs, these would go through the dorsal ramus of the spinal cord, right? So you can see this, remember that this is on the dorsum, and this is the dorsal aspect of the horns. And this would go through, and then it would enter the dorsal white column, either the gracile fasciculus or the cuneate fasciculus, and these would then ascend, still here, into the gracile nucleus and cuneate nucleus. So why do I call it a nucleus? Okay, that's my Facebook. Why do we call it a nucleus? Because it has a collection of cell bodies. Cell bodies from where, you guys might be asking? Well, cell bodies from the next bit, which is where the second neuron, it starts there, and that's why you have cell bodies that are collected from the second nucleus. If my friend would just stop texting me. These cell bodies are collected from the second nucleus. They start at the gracile and cuneate nuclei, and they go into the medial lemniscus on the opposite side of the medulla. So this is known as decussation, when it basically crosses from, well, I can't really tell if this is left or right, but you can see it goes from the left side of the picture to the right side of the picture, and that's just a fancy term, the fancy term we used to say crossing over. Okay, so it decussates over to the medial lemniscus on the other side of the medulla, and then it further ascends all the way into the thalamus, where it synapses once again. And that's when our third neuron takes over. The third neuron basically goes from the thalamus to the postcentral gyrus, and that's when we process our sensation. So in this case, it will be, oh, I've been tickled by a feather. Okay. And then the spinothalamic tract. This is going to be a little bit easier just because it's less complicated. Basically, what happens is we go from sensory receptors, again, pain, temperature, and crude touch. These go up to Lysau's fasciculus and the lateral/ventral spinothalamic tract, medulla oblongata, pons, midbrain thalamus, and then finally the postcentral gyrus. So you can see the first neuron goes from right here in the sensory receptors all the way up to the what I believe would be the lateral/ventral spinothalamic tract. This would go all the way up once again to the thalamus. This should be at the thalamus. And then from the thalamus, the third neuron goes all the way up to the postcentral gyrus. Okay, so let's see here. So when something when you touch something like fire or a hot plate, this is pain, and this is also quite a lot of temperature, I would say. So this first neuron detects that, and then it goes all the way up through once again the dorsal horn, remembering that the dorsal horn takes care of afferent neurons. It takes care of sensation, while the ventral one over here would always take care of the motor neurons. So it goes through here, the dorsal area, the dorsal horn, and it goes, the dorsal root, and then it goes into Lysau's fasciculus and its synapses right here. Okay. And then next, from Lysau's fasciculus, these neurons would then decussate. So decussate, crossing over to the other side of the spinal cord. So here it would end up in the lateral spinothalamic tract. From then it ascends into the thalamus. So as you can see here, there's no involvement of the medulla, and that's why we call it the spinothalamic tract, because it goes, as you can see, from the spinal cord right here into the thalamus without the involvement of the medulla. I mean, obviously it still goes through the medulla, but there's no synapsing and there's no decussation that happens inside the medulla, so we just kind of skip out on that entirely. So it's the spinothalamic tract, okay? And lastly, our third order neuron takes us from the thalamus to the postcentral gyrus, where we rightfully interpret the signals from putting your hand on fire as okay, and now lastly, the corticospinal tract. So contrary to the previous two spinal pathways, the corticospinal tract only has 2 neurons and it only synapses once, as you can see right here. So the corticospinal tract takes us from the precentral gyrus. So contrary to the previous 2 tracts, the postcentral gyrus is the one that takes care of the sensory information. The precentral gyrus takes care of motor information. So we are taken from the precentral gyrus all the way to the lateral and anterior corticospinal tracts in the medulla, the lateral anterior corticospinal tracts in the spinal cord. This would be roughly when the upper motor neuron synapses with the lower motor neuron, and then that's when we get to the motor receptors on muscles or glands, okay? So first, this looks a little bit complicated, don't worry. So we have the postcentral gyrus right up here. It goes down. In the case of 90% of the fibers, they decussate really early. They decussate right in the medulla, from the medullary pyramids all the way across to the other side of the medullary pyramids, and then this will take them into descending into the lateral corticospinal tract. Okay, so remember that if it's lateral, it means it has decussated really, really early on in the medulla. Now, what happens to the remaining 10% of these upper motor neurons? These would go from the precentral gyrus into the medullary pyramids. They wouldn't decussate right here. Nothing happens to them. They keep descending into the ventral corticospinal tract. And then at this point, then they would decussate into the ventral corticospinal tract. So you just remember that lateral means it decussates in the medulla, ventral means it decussates in the spinal cord. Okay, yep. And then next up, we just have a very simple path from the lateral or the ventral corticospinal tracts that go out through the ventral root, and they go straight into your into the muscles in your hands. For example, when you're smashing the subscribe button on the PS Pain Relief YouTube channel. Woo! All right. I do not like the way I did that. So yeah, by the way, if you like our stuff, please subscribe to us. We need the ad revenue. And next, okay, so reflexes. Reflexes are much faster in nature than our spinal tracts. Okay, you can just see right here, there are 2 types, monosynaptic or polysynaptic. It's kind of self-explanatory. So monosynaptic reflexes just have 1 synapse right here. Polysynaptic reflexes have 2 synapses and there is an interneuron involved, and this would be slightly slower than polysynaptic reflexes. Okay, and last but not least, divisions of the autonomic nervous system. So I've talked to you guys through ascending and descending pathways. So, but for descending, I think we've only touched on somatic motor pathways because those are the ones that let you do voluntary movements. But what about visceral motor pathways? Well, these are the ones that regulate involuntary responses in smooth muscles, cardiac muscles, and glands. So this would fall under your sympathetic and parasympathetic nervous systems. So parasympathetic versus sympathetic nervous systems, what are they? So parasympathetic basically just means the responses that your body has when you are at rest, when you are completely chilled out and when you're not running for your life, versus sympathetic responses, which are what happens when you're in a fight or flight situation, or you're just in a very, very tense kind of state of mind. And you can kind of logic it out in such a way, as, you know, let's say if you were running for your life, what do you think would come in handy in terms of your physiological responses? So your pupils will need to dilate because you would want to see the maximum amount of information to kind of decide if you have to dodge something. Okay, and your salivary glands, you wouldn't be salivating at this point. I think your mouth would just go completely dry. I mean, you don't want to lose that kind of extra water and extra ions, I guess, through your mouth. Your heart, you would accelerate your heartbeat and you would dilate your bronchi, and that's because you want to get the maximum amount of oxygen through your blood to your muscles to help you keep running and keep on the move, keep being on the move. And your stomach would inhibit digestion, while your liver would stimulate glucose release, because again, you want the maximum rate of respiration happening inside your muscles, inside your little mitochondria to help you keep moving. Your kidneys would release adrenaline and noradrenaline, and that's to keep up the sympathetic response, as we'll find out later on. And intestines will inhibit peristalsis, just like the stomach. There will be less gut motility, and that's because you're not exactly going to be munching on a Kit Kat bar as you're running for your life, right? So you would probably not need to digest anything. And lastly, your bladder would relax. Yep. And sorry, I moved on too fast. And once you kind of logic out what you do want to be experiencing when you're running for your life, that's when you can kind of decide, okay, the flip side would be true. For if I'm just kind of laying down on my bed playing with my phone and just chilling out, you know, with a bag of chips or popcorn. I have popcorn right there. Anyway, different neurotransmitters and receptors. So as we know, neurons have to synapse, right? So for these autonomic nervous system neurons, they would synapse right in the in ganglia. Okay, so so the ganglia, which basically just means a collection of cell bodies in like a synapse. This would mean that the sympathetic nerves would synapse in the sympathetic ganglion, which, as you can see, the moment they escape from the spinal cord, they would kind of form this little bulbous looking chain right beside the spinal cord, and this is where your sympathetic nerves are synapsing. So you can see here that, as you can imagine, because the preganglionic neuron synapses so early on, the postganglionic neuron is going to be much longer than the preganglionic neuron. Whereas for the parasympathetic nerves, they would synapse right on the target organ itself, and there would only be a small little kind of ganglionic cell on the surface of the organ itself. So you can see that the preganglionic neuron is very long, while the postganglionic neuron is just really, really short. And yeah, this would be the target organ. So the neurotransmitters in the parasympathetic versus the sympathetic system. So for preganglionic, it's acetylcholine for both, and these acetylcholine neurotransmitter molecules, they would attach to nicotinic acetylcholine receptors. Okay, so nicotinic receptors, and that's common for both preganglionic neurons from both para and sympathetic nervous systems. So postganglionic, for parasympathetic, we would still keep on using the acetylcholine neurotransmitter, and then the postganglionic receptor, so the receptor that's right on the target organ, that would be therefore a muscarinic receptor. Okay, it's still using acetylcholine, except that instead of being nicotinic, it's now muscarinic. But then for sympathetic, as you can guess, we're going to be using adrenaline instead, noradrenaline, right? And simply put, the receptor for that is adrenergic. So it's the adrenaline binding to the adrenergic receptor that's on the surface of the target organ itself. So next up, types of receptors. So for muscarinic receptors, we really only need to know the M1, M2, and M3. So M1 takes care of salivary glands, gastrointestinal tract, and mucociliary cells, particularly the ones in your trachea. And then M2 would be cardiac muscle, M3 would be salivary glands, and smooth muscle, again, mostly in the airway. That's why when you have asthma, the drugs that you would use to treat asthma would be targeting M1 and M3, because both of them take care of things that are inside your trachea, inside your airways. And nicotinic receptors, well, there are two subtypes, but honestly, you guys don't need to know that. So just know M1, M2, M3. And then for adrenergic receptors, we have A1 and A2, alpha 1 and alpha 2. Alpha 1 takes care of arterioles, veins, and iris muscles. And alpha 2 takes care of your salivary glands and gastrointestinal tract. And the one that you really need to remember would be the beta receptors. So the beta receptors, you have beta 1 and beta 2. So beta 1 takes care of your cardiac muscle, most importantly, and your beta 2 takes care of your bronchi, which also one of the more important things you need to know. So how do you remember what beta 1 and beta 2 do? You just remember that you have one heart and two lungs. And fun fact, this is a picture that came up when I searched B1 and B2 without also searching receptors. So, oh well, anyway. But that does it for this installment of PS Pain Relief. Thank you guys for sticking around. Sorry we didn't get to you guys last week, but we hope that this has been helpful. Once again, if you guys have any questions, please drop them into our personal Facebook inboxes, and we'll try and get back to you as soon as possible. Don't hesitate to give us feedback, to give us questions, or just to tell us, what worked and what didn't. So I'll see you guys next week. Bye.